happy. This is the last of the fall semester of the PowerTalks series. I think looking back, we've been running this series for over 6 years, achieving various impact. And if you all remember, Dr. Jim Semino's talk attracted more than 100 persons from all over UAB and throughout the regions. Certainly, we're going strong next semester with speakers from both UAB internal and outside of UAB lining up. If you are interested in presenting and haven't done so, please contact Amy Wang. Okay, with that advertisement, I'm very happy to introduce our speaker today, Dr. Jing Su. Dr. Jing Su is currently an assistant professor at the Department of Biostatistics and Health Data Science at the Indiana University School of Medicine. He certainly have a very unusual but long productive career in the engineering flavored bioinformatics and systems biology. I'm emphasizing this because that I'm looking at his trajectory, he almost perfectly fit our new design of a Ph.D. program in biomedical engineering with a concentration in bioinformatics, which is brand new, and it's going to be launched by the end of this month to attract students. He actually got his bachelor degree from Tianjin University in material science, but coming to the United States, he pursued a PhD at Georgia Tech in biomedical engineering. And how he ventured into bioinformatics, he was through a lot of the interesting, maybe mentors and colleagues. I think in 2009, upon his graduation, he went to University of Massachusetts Amherst to investigate network biology and maybe circadian control of cell cycle. And then he has lots of encounters with clinical and biomedical informatics organizations. He worked in the Methodist Hospital Research Institute and Weill Cornell Medical Center of Cornell University at Houston. And then he had worked also in the Wake Forest University in the Wake Health from 2013 up until he joined Indiana University. His research is very, very broad, and that already transcends beyond the material science, biomedical engineering, to include many aspects of biomedical informatics, machine learning, data mining, immunology-based systems biology. And I'm pretty sure that he has something interesting and really avant-garde to say today. Without further ado, let's welcome.
Okay. Thank you, Jake, for the invitation, for the great introduction. So today I would like to share our recent progress in using graph artificial intelligence to review health trajectories for chronic kidney disease patients. And of course, from the very beginning, I would like to give an overview of our research domain and our trajectory of our lab in research. So first of all, I serve as the PI of our biomedical informatics lab and assistant professor in our department. And meanwhile, I also serve as a director for the data management services team. We have a team of 27 members, and with an annual budget over 2 million, and we are supporting over 100 projects. Also, I served as the associate director of real world for the biostatistics and data management core at IU Comprehensive Cancer Center. So my team in Cancer Center is supporting the research use of clinically generated data, including medical claims, medical records, and multi-omics genomics data and imaging data. So for research in my lab, currently we are focusing on developing and implementing graph artificial intelligence and other graph-based algorithms on longitudinal real-world data and multimodal biomedical big data to study chronic and complex diseases such as chronic kidney disease, cancers, and their interactions with COVID for better and precision care of those patients. So because this talk is focusing on graph approach, so I would like to outline my lab trajectory in this direction. One of the early publications from my lab is back to 2015. So it's focusing on using relational dependency network to incorporate the clinical data and the clinically generated genomics data collected from Wake Forest to better understand the breast cancer patient subtypes. So we published a paper on scientific reports. The basic idea is trying to build up the discovery clusters of the clinical and genomic features, and using those clusters to better identify subpopulations of breast cancers that they may show a unique molecular mechanism and show unique survival and treatment responses to treatment. And later on in 2018, collaborating with Dr. Li Wang from UT Arlington, we prepared a study using the reverse graph embedding approach to identify trajectories of chronic kidney disease patients using both the electronic medical records and the genotyping data generated from a series of clinical studies at Wake Forest. And then we are able to identify trajectories of different risks and different progression patterns. And specifically, we are able to identify trajectories that are dominated by specific genotypes, especially the famous kidney risk genotype APOL1. And this approach helps us to better predict the patient's trajectory. For example, if you look at the left side, along the green curve, you can see that the kidney functions of these patients was damaged at before age 45 and quickly recovered. And then clinically, we don't know whether the patient's recovery is a full recovery or it's just transient. And then later on, at age 46, the patient's kidney function quickly declined and developed kidney failure. So currently, it's challenging clinically to predict whose recovery will be permanent, whose recovery will be transient. But through deep learning approach, we can find out that these patients, although they appear to have a recovery from the clinical indicators, but actually in the latent space, they already switched to a different trajectory, the risk and fast development trajectory, so that it allows us to use the full scope of clinical data and genomics data to better predict the patient's trajectory that provide a better support for clinical decisions for clinicians. And then starting in year 2019, and my lab moved more into graph AI. So we start to publish papers in this domain in 2021. For example, that we first published, I think it's one of the first paper using a graph convolutional network to incorporate both clinical medical records and the genomics data generated from foundation medicine to better understand the different responses of non-small cell lung cancer patients to immunotherapies. This approach is for the first time allowing us to incorporate both data and using a graph AI to predict who will respond better to immunotherapy, and also to reveal underlying molecular mechanisms, which mutations and copy number variations might be responsible for better or worse response to the immunotherapy. And this also casts new lights on the existing biomarkers used clinically. This published on Nature Digital Medicine, and then later on collaborating with Dr. Qianqian Song at Wake Forest, we published one of the first, if not the first, graph AI model in single cell domain to do knowledge transfer from reference single cell RNA-seq data to different type of single cell omics data, including single cell RNA-seq data, RTEX data, cross samples, cross platforms, cross different type of tissues, and cross species. This paper is published on Nature Communications, and later on we published one of the first graph AI model to use the single cell RNA-seq data to deconvolute spatial transcriptomics data, also using the GCN model. This is also collaboration with Dr. Qianqian Song. And then later on, we move to the single cell spatial transcriptomic data that are emerging since last November. And now we can see more and more attentions are focusing on this new commercial technology. And I think that we are one of the first that use the single cell spatial transcriptomic data, especially the nanostrings cosmics data and the merge scopes and the base genes merge scope data to analyze and to learn the spatial domains and to learn cell-cell communications. So this is also collaboration with Dr. Qianqian Song at Wake Forest. So we are using graph transformers and adaptive graph learning model to handle this single cell spatial data. So, and also I've been working a lot on other real world data to generate real world evidence for clinical decision support. And I have been collaborating intensively with Dr. Noha Sharafeldin at UAB. So we co-published 2 GCO papers. The first one, Noha is the first author and the corresponding author, focusing on the outcome of COVID-19 patients who have cancers. And then this year we published another GCO paper that the collaborating with Dr. Qianqian Song and Noha, also on GCO about the breakthrough infections in cancer patients after they received the COVID vaccines. So let me go directly to today's topic on the how to use graph AI to investigate longitudinal real-world data, including both omics data and the clinical data, to better address the challenges in precision care of patients with chronic diseases. So first of all, a little bit introduction about the data that my lab currently is focusing on. The first one is the Optum data. So these are medical claims of the privileged population. So currently we have about 75 million patients since 2007. And the current computational environment is on cloud. It's called the Optum Cloud. It's a evidence-based cloud environment. Another data is all of us data is on Google Cloud. It's focusing on underserved populations. It has the electronic medical records, genomics data, and a rich series of surveys. And also, of course, our local electronic medical records called the INPC data. INPC data collect the electronic medical records from hospitals located in Indiana. It covers over 2/3 of Indiana population, and the data traces back to 1995. So it has our regional electronic medical records, multi-omics data generated in clinical and in research, imaging data, and also have rich biospecimens, which is also very important if we want to do validation or generating new data. We can use the biospecimen collected in INPC data. And of course, we have a national collaborations in the N3C to better understand the interaction between other diseases and COVID. So the NOHA, I, and a few colleagues are co-leading the cancer domain team for N3C. So that's the data used focusing by my lab. Then I would like to introduce chronic diseases. This is sometimes called the non-communicable chronic disease nexus. So it has a group of diseases share similar risk factors, including cancers, diabetes, cardiovascular disease, chronic kidney disease, cognitive impairment, including Alzheimer's and dementia, COPD, and other diseases. These diseases are sharing similar risk factors. Number one, of course, is aging, and also it has a lifestyle impact, and social economic determinants play a major role, and also related with other clinical conditions such as obesity, metabolic symptom, and chronic inflammation. Genotypes also play a major role in here. And among 6 in 10 adults have at least one of these diseases, and is responsible for over 90% of annual cost in medical care, and is a #1 cause of death, of course. And even you separate them, they are dominating the top 10 causes of death. And luckily, many of those conditions are preventable or manageable. And these diseases are often the comorbidity of each other's. If you are focusing on one of them, very likely that you are working on all of them as comorbidities. And so there are challenges in studying chronic diseases. First of all, just I mentioned that there are so many different type of factors impacting those diseases. So these diseases are highly heterogeneous. So that we need a large sample size to cover smaller subtypes. And also we need a large sample size to cover the different factors that impact the diseases, and also to cover, better represent the whole general population, and also the heterogeneities among populations across different geographic regions, across different hospitals and providers, also play as a major confounder of these diseases. So we really need a large sample size, and also need a very data with very good representativeness. And also because so many features are impacting these diseases, so that we need the rich features across multiple domains, from clinical, demographic, socioeconomic to genomic domains. And of course, those are chronic diseases that take decades to develop. Longitudinal coverage of the data is important. And because this disease is so complex, so usually it requires a multidisciplinary, multi-institute collaborations with domain experts in bioinformatics, biostatistics, bioinformation, data science, clinical care, nursing, clinical trials, and so on. And also we need a lot of IT support in addressing the data standardization, data engineering. So there are so many challenges in studying these chronic diseases. Then let's look at the data part. So think about what the ideal data will look like. So if we look at data from three perspectives, at the longitudinal dimension, we can define such data as its length. And at the patient level, cohort level, we call it the breadth of the data. And at the feature level, we call it the depth of data. The ideal data will be that you have a giant population, you have the follow-up of the population over all the time, and you have the data available for each feature. That's the ideal data. So how about the clinical data look like? Data that are collected for clinical studies, it has a much smaller cohort, have a regular follow-up, and focusing on a handset of the features. The good thing is that the follow-up is quite regular and you have quite a good data for the features collected. The issue is that the coverage of the cohort and the features are very limited for studying such complex diseases. And of course, the real-world data is promising. It has very good coverage on time. It often covers decades. You can often be able to trace back to 2000 or even in 1990s, 1980s. Have very good coverage of patients, and the common data cohorts cover dozens of millions of patients. For example, Optum covers 75 million patients, and the INPC covers 10 million patients, and also the has a rich coverage of the features and the across multiple domains. However, when you look at every patient, the data are so fragmental, have very limited coverage and completeness of the data. So here I use a few examples to further emphasize the challenges and the unique features of the data. This is the non-small cell lung cancer patients provided by Flatiron, which is I think the #1 data company. It's through the collaboration with AstraZeneca. On the left side rows are features. You can see both molecular features, genomic features, and the clinical features. Columns are patients. Blue parts are available features. Gray parts are available features. We already focused on the most rich features. You can see that we are still missing a lot of features. And then the missing of features are strongly correlated. And the availability of features are strongly associated with the clinical conditions. So we call it informative censoring and structurally data availability. Informative censoring means that availability of the feature reflects clinical conditions. Structured availability means that the availability patterns are strongly associated across different features. For such features, it's so hard to impute them. And also you can see on the right side, those features show strong associations. So from the advantage part, if you are missing some features, information can be provided by other features. But on the downside, the many statistical models suffer from this strong association cross features. And this paper, in the paper published in 2016 by our team, we try to study whether we can impute some features. We realized that for many features, if we use the modern imputation technology, actually we generate artifacts that actually contaminated the downstream analysis. On the left side, you can see that from the real data, which is the black spot, we can find that association between the B cell, plasma cell enrichment, and the tumor size are not strongly associated. However, after imputation, we can find that they are strongly associated. And a little bit of diagnosis into the imputation process shows that we are actually generated artifacts of the strong association between B cell enrichment and tumor size. So it shows that the traditional imputation method may bring in lots of artifacts for downstream analysis for such type of data. And here I would like to further demonstrate the concept of informative accessory. This is a KM curve, survival curve for the risk of developing cardiovascular disease after COVID infection. And then we group patients with different kidney functions. You can see that for patients who are missing the data, which is the red curve, and for patients who have healthy kidney functions, they have similar risk, which means that the missing of the kidney function data actually is an indicator of good kidney health. But of course, it makes sense that they don't have data because clinicians don't think that the necessity of collecting that data. And it's also shown here that about whether the troponin I level can predict a patient's risk of developing cardiovascular events after they get COVID. You can see that patients who don't have the measurements actually have the least risk, which is reflected by the red curve here. It even have less risk than patients who have normal troponin I level. That's largely because that troponin I test is often only ordered by clinicians when they have concerns about the cardiovascular damages. So that's why. So here, I hope I have demonstrated that the data availability pattern sometimes even play a stronger, provide stronger information about the clinical conditions than the actual value of a feature. Okay. So in our specific studies, we can see more detailed challenges. For example, when we are working on different data resources, we are actually focusing on different population. When we're working on the Optum data, we are focusing on the clinically privileged patients who have a commercial health insurance. When we are working on N3C data, then we are looking into populations that the big research hospitals see. And also there are reporting bias that Noha and I and Dr. Qian Song have been studied a lot in the N3C data. There are different type of health disparity involved in different data sources. This all bringing so many confounding effects when analyze data. And also it's very hard to distinguish between confounders, the indicators or the markers, and the real causes in analyzing real-world data. And also the data continuum is a big challenge when we work with real-world data, because one patient may see multiple providers. And if we only look into the data by one provider, maybe we are missing a lot of data. For example, the cancer patient diagnosis is often in smaller hospital, and once the case gets worse, they transfer it to bigger cancer centers. So we may not have the initial genomics data and clinical data about cancer patients, and we see more patients with relapse cases that challenge us a lot. And of course, we have the multi-modality data, across the genomics, clinical imaging, and other domains. That is also a challenge when working with the real-world data. So the graph AI-based latent representation is promising in addressing some of those challenges. First of all, by using graph representation of the real-world data, we can easily capture the informatively sensitive pattern by introducing specific features to capture the availability of those features. And it's very easy to incorporate data from different domains because the graph can easily integrate such data together, as we have demonstrated in our previous work in incorporating genomics data and clinical data. And also it allows us to incorporate knowledges from known relations also as a graph. And also the latent representation can bridge the gap between the very noisy, heterogeneous raw data seeing the real world scenarios to the existing models. Once we use a graph AI approach to project this noisy, heterogeneous data into a latent space, the mathematical properties get much better, and many latent features by optimizing the model can be relatively independent, continuous, and differentiable. That allow many statistical and machine learning model to be used here. I will show some examples in the following slides. And it allows us to capture common data patterns and removing bias. As well known that once we project the original high-dimensional heterogeneous data into a relatively lower dimension in latent space, many biases are removed, noises are canceled out. And it's versatile for different applications like supervised, semi-supervised, unsupervised learning, domain transfer, knowledge transfer. So it's quite versatile for different type of applications. And also it's compatible with existing statistical and machine learning models. I will show you how it works. So I would like to use our recently funded R1 project to demonstrate what I discussed so far. This is a collaboration with Dr. Chen Song from Wake Forest. Our recently funded project is called the DPORT, the Disease Progression Trajectory, an evidence-driven graph-based clinical informatics approach. So this specific work is focusing on how to better control the risk of drug-induced acute kidney injury among patients with chronic kidney diseases. So first of all, of course, CKD is a heterogeneous disease. It's underlined by different pathological and molecular mechanisms, show different age-related comorbidities, and reflect different impact with social determinant of health, lifestyles, and health disparity, and the genotypes, especially risk genotypes, also play a very important role here. So it needs the incorporation of the genomics and the clinical data together. And also among the chronic disease patients, because they are often senior people, they often have multiple comorbidities, so they often use a large amount of drugs together for long term. That dramatically increases the risk of drug-induced acute kidney injury. This is a major threat to the health of chronic kidney disease patients. However, it's very challenging to analyze that because the population of the chronic kidney disease is so heterogeneous, and the use of drug is so complex. So we rely on big data and to identify specific populations of patients and the specific drug combinations to better understand the risk. So the motivation will be that we observe the fragmental data from patients. On the top, each line is the longitudinal visits of a patient. We can see that they have very fragmental data. The idea is that if we can assemble those fragmental data to learn the underlying trajectories of those patients, and then we may be able to analyze the specific risk of the patients in different trajectories and at different locations of the development status at each trajectory. This may allow us to identify which drugs are safe for some patients, but may cause kidney failure for other patients. So, the basic idea is to use the graph representation to represent the longitudinal real-world data from the cohort, including both their clinical and genomic information, and to represent demonstrate here their longitudinal patterns as a graph, their clinical and genomic features as the attributes of each node. So here each node represents an encounter, and the edge represents the similar age between two encounters across the whole population. And then using an inductive graph AI model, here we use the graph stage, we can project the patients, the visits of those patients into a latent space. And then in the latent space, we can use the machine learning models, such as reverse graph embedding models, to learn trajectories. And then on each trajectory, we can further analyze the patient's risk of kidney failure and the interaction of patient's kidney disease and other diseases. Demonstrated here are the interaction between patients of different kidney disease progression trajectory and different cancer types. So, and of course, it can be expanded to other comorbidities. So, a quick the introduction on the design of the study. The cohort is from Wake Forest Hospital. We have over 1.6 million patients, and after screening, for those who have diabetes disease and have enough measurements of serum creatinine data, we generated a study cohort of about 80,000 patients that we have the longitudinal measurements of kidney functions. And this has covered about 1 million kidney function measurements called the EHR data. And then we separate these patients to 2 cohorts. One is the learning cohort, one is the prediction cohort randomly. Then we are using the learning cohort to train the graph AI model to project any patients into the latent space. They use the prediction cohort to project the right patients and their data into latent space. And among them, a significant amount of them, we have the genotype information available. And here, I would like to show the latent representation after the projection of the raw data into the latent space. Each dot here is a clinical visit, and we are showing the latent space by UMAP visualization. And for the top one, we visualize the visits by age and color-coded the senior the visits from the senior population in red and the younger population in blue. You can see that the latent representation after graph AI projection can capture the pattern of age. And on the bottom, you can see that we can capture the progression of kidney disease The red part means that the kidney deficiency. Blue part means that the healthy kidney function. You can see that we can capture the pattern of the kidney disease progression. And on the right side, we use a reverse graph embedding to generate the trajectory. For those who are not familiar with reverse graph embedding, if you are familiar with the trajectory analysis, Monaco and other trajectory analysis approach developed for analyzing single-cell RNA-seq data, that's actually the same model. So we just use a bioinformatics model to study the clinical data. So you can see that on the trajectory we developed, we can capture the age pattern, which means that we can capture the aging process along the discovered trajectory. On the bottom, you can see that we can capture the progression of kidney disease on the trajectory discovered. So this slide shows that we can, after latent space representation, we captured the pattern of aging and the pattern of progression of the kidney disease. And on the learner trajectory, we can also capture both of them. And further, I would like to show you that, sorry, there's a little bit of mislabeling on the left side. Don't worry about that. Further, we show that on the top, we can capture the difference in the race. The black people are highlighted as yellow and white people are highlighted as blue. You can see some trajectories are dominated by African Americans, some are by non-African Americans. And at the bottom are capturing about the difference of sex. You can see that some trajectories are dominated by female, some by male. And both those demographic informations are also well captured in latent space. Then I would like to provide a little bit of the background knowledge about what a trajectory looks like before I explain the further the results from our discovery. For those who are familiar with trajectory analysis in single cell, maybe it's already very familiar, you already know what I'm talking about. So for a learner trajectory, it's actually represented as a directional chain, which is a specific type of graph. So each node, we call it a landmark, represent a group of clinical visits, and the edge between two nodes represent the kidney declining from one stage to next stage. So each node represents a group of the clinical visits, and the edge represents the longitudinal progression of the disease. Okay, now let's look at the left side. For each node, we visualize with a unique color. So each part with each section can be understood as one node, and then the each nodes are linked one by one along the trajectory. In the middle, we show the distribution of the age associated with each cluster. You can see the beautiful capturing of the trajectory of the aging process from those landmarks. On the right, you can see the diversity of the kidney functions through the aging. For the each row represent the same cluster of both age and EHR functions. So you can see that when patients are young, most of them have very healthy kidney functions, and during aging, we start to see the some patients still have good kidney function. Some patients start to develop the kidney injury, and then we try to annotate the discovery with cancers, different type of cancers, use TF-IDF enrichment analysis. Then we start to find that some cancers are strongly and positively or negatively associated with kidney disease progression. And I will show you specifically. And we can see that we can classify cancers to three groups. Some cancers are strongly associated with severe kidney damage. Some are associated with healthy kidney function. Some are associated with mild injury of kidney functions. So on the left side, we show the raw enrichment results, and on the left part, those are patients with kidney issues. Right part, along the right part, right section, are patients with healthy kidney functions, and the association strengths with different kidney function status are color-coded. Red color means the strong association. And after adjusted for age, sex, and race, we still see similar pattern here. And then I will move into a specific cancer type to give more insight into our discovery. For example, brain tumor associated with very different kidney trajectory and it's associated with a healthier kidney function than the prostate cancer and the melanoma cancer. You can see different enrichment of different cancer types. And then we further focus on two trajectories showing us up and down. Then you can see that these are covering patients of similar age group, but with very different the kidney function trajectory. And if you look further into it, comparing with the overall population, the one sub-trajectory have much healthier kidney function, the other have very bad kidney functions. And this is true both in our learning cohort and in our prediction cohort. And then we further analyze that whether some diseases are associated with them. We find that, okay, the breast cancer are strongly associated with a low risk trajectory, and both in learning and the prediction cohort. And we further show that for those rare cancers, we may not see clear pattern in the learning cohort, but once we use graph AI to project the whole population into the latent space, now we can see strong and clear pattern of which cohort, which trajectory they are associated with. So we are almost at the top of time. So I would like to summarize what I have discussed today. So hopefully I have shown you that the real world data have very goodly cover the longitudinal range and cover multiple data domain, cover a giant cohort, and it has provided rich data in genomics information, in clinical information, in imaging information, and demographic information. How to effectively use such fragmental and rich real-world data to study chronic disease and to provide precision medicine is a major challenge here. Our team built up graph AI model to address the challenges of using longitudinal multi-model biomedical data for precision medicine for the chronic diseases. And I have shown here that our model allow us to do trajectory learning, and which provide a foundation for precision medicine, allowing for the first time to separate the patients to different trajectories, then focus on their progression phase on each trajectory to further analyze what's the risk and and how to better care for them. And what I have not shown here is that, for example, in the DEEPENS, we do the regular k-means clustering in latent space to identify those patients that respond differently to the immunotherapy. And in our follow-up papers, we use a different clustering approach in the latent space to identify which cells are communicating with each other, to identify spatial domains that have different functions in cancer and in healthy tissue, and to also to identify the other interesting information. But we are using a similar mechanism strategy, projecting heterogeneous data using a graph AI into latent space. Then from there, we can start our analysis using traditional method to get better understanding of the heterogeneity of the problem and to generate actionable knowledge for precision medicine. And so we are actively working on both the clinical questions and the bioinformatics questions using the same approach. So of course, at the end, let's say that we are looking for postdocs that are interested in graph AI and biomedical informatics. And also I'm hiring master level data engineers and data analytics into our team. And I would like to thank to our collaborators and our team members. So questions?
Well, thank you so much, Dr. Su. And I find your innovative use of a trajectory analysis coupled with a graph embedding to be quite inspirational, because the people normally previously, I only see them in a single cell analysis, and you really apply it in the precision medicine context. Maybe I'll start with some questions. So one is, I noticed that you use a three-dimensional projection embedding more extensively, whereas other people use two-dimensional. Do you truly see that three-dimensional add a lot of resolution, or the 2D is equivalent? That's the first thing. Yeah. And the second is you mentioned that that you're aiming to do causal analysis. I wondered if this real-world data, graph AI, and trajectory analysis still are primitive in getting you only the candidate association, and the causal would still, you have to work with clinicians, or you think that we are already at the stage of using informatics to do causal, causal actually finding already.
Yeah, great questions. So first of all, the this trajectory projection to 3D space is based on collaboration with Dr. Li Wang, who is the one of the major author of the original graph embedding, reverse embedding paper published on Nature Method for the single cell data. So according to her, she mentioned that the 3D space allow all possible topology to be learned and visualized for a graph. And 2D, many edges may overlap. 4D, not necessary. So 3D, so there's a graph feature for 3D space. It allows us to visualize any possible graph topology. So that's why we use a 3D. And the second question that we are using clinical design into the real world data, try to better distinguish confounders and the real causes. However, this only help a little bit, and we can generate more reliable hypothesis, and such hypothesis still need to be tested in clinical trials and clinical studies. And one major way to do it is through pragmatic trials. So you can leverage the electronic medical records and the clinical care pipelines to do that one. But still, I think clinical trial is necessary. And also I see a question from the audience. It's talking about whether the CKD, different association of CKD and cancer are associated with the specific treatment. Yes, exactly. That's exactly our hypothesis. We want to know two things actually. One is that the whether kidney functions limit the choices of cancer treatments because of the toxicity. Second, whether some treatments are more likely to trigger specific kidney damage. And also, not mentioned here is the confounding factors here. And for example, the association with some cancers with healthy kidney function usually means that poor survival of the cancer, so that the patient didn't get a chance to develop kidney disease. So yeah. For embedding, the next question, did you do any customization of the graph neural network to better suit for longitudinal data? Yes. Thanks for the question. Yes, we did a lot of pre-processing to capture, for example, the missing data, to avoid the imputed data to dominate the study, to capture the longitudinal intervals, and also this is actually one of the major focus now for us to better learn the longitudinal trajectory in latent space by regulating the study by some loss functions. Yes, a lot of details there in order to make it work. Yes. For patients with AKI, COVID-generated or drug therapies, once they get to the ICU, it's too late. The injury is established and there is limited therapy. Can you project whether new parameter can be identified as a predict AKI before it is irreversible? Yes, that's a great question. To our model allow us to predict the relative AKI risk along the trajectory, and currently we are specifically interested in risky drug interactions that may cause induce AKI. This can be prevented for specific patients. So that's one of the major focus to avoid giving patients the risky drugs that are still unknown so far. So we want to provide that knowledge.
I know that there will be more questions, but we can write to you, I presume. Your work is really amazing, and we thank you for your time spent with us this morning. With that,
sure.
Yeah, we'll give you a virtual round of applause. Thank you.
Okay, thank you. Really nice to talk with your audience.
Okay, all right. Now we're done, and see you in this seminar series next year. All right, bye.
Happy New Year, everyone.
Mm-hmm.